repeat sustains, because this is what happens when someone calls me in another center and says, we have a patient who has X, Y, and Z, and they want to do A and B. Can we do that? And I have to thumb through the protocol and say, what were the rules? It's really helpful to have a protocol that's consistent. If you're writing all of your protocols in your own place and nobody else is involved, just use your same outline every time. If all of your protocols are phase 3 NINDS funded trials, congratulations, first of all. And second of all, they will tell you what your protocol will look like, and there's no room for that. If all of your protocols are written with Pfizer, they will tell you what they want their protocol to look like. That's somewhat negotiable. So I'm going to literally go page by page of what has to be in your protocol, because what you should do tonight is go look on that disc or on these websites of the protocols and start thinking about what's going to be in your protocol, because on Wednesday you're going to hand that to your team that's going to review that for you, and you need to start filling in those gaps right this second. So you need a title, a protocol number. That's going to depend on your institution and also the institutional support. You need your name. How are they going to get in touch with you? When something goes terribly wrong, how are they going to page you in the middle of the night to tell you there's been a problem and that you need to address it? And then all these things on the side are optional, and they're in your slides. Then here are the basics, the subsections, and these are arbitrary, but really the point is to make sure, again, for yourself, that we've made a listing of everything we have to think about before we expose a patient to risk. What are all of the things we have to consider and detail? And what I like to say is a protocol should run so that if tomorrow I decided to raise sheep and make goat's milk, that the protocol could carry on exactly the way it had started 5 years ago without any deviation. And that's what your protocol should allow you to do, that people can run it on its own. So this first, right after the title page, is often a summary page, and this is an example of one out of my protocols, and this is literally one page saying everything I'm going to do in the whole protocol. And you're not meant to read this, but my point is that it's a summary of the highlights, so that if you're trying to read something very quickly, or the IRB is trying to read something very quickly, they can read this one page. A schema is optional, but very helpful. If you're in my group, I often ask for a schema as the very first thing that we do, because it requires clarity. If you can put it into one figure, then you understand exactly what you're doing. And so this is also very helpful to have in your protocol document, because you can refer back to it in a heartbeat and understand what it is that the whole schema is. So you can see this one schema represents the entire study from enrollment to the off-study time point. Here's another schema. This is one that I often ask people to make as we're initially talking about a protocol, the concept. This would be a great one to think about making tonight as you're starting to populate your protocol. What are the basic requirements? What's your eligibility? What is your primary endpoint? What are you going to do? What's the process of the intervention? And this should, again, be one page. I don't know how many words this is, less than 100 words. Your objectives are really the meat of the whole protocol. This is what is the question you're asking, and then everything else will stem from that. This sounds absurd, but when I say that the primary objective has to be consistent throughout the protocol, I can't tell you how many times I sit on the Data Safety Monitoring Review Board for the Cancer Center, and I sit on the Cancer Review Committee for our protocols, and I can't tell you how often the stated objective is different in 5 different places in the protocol. And that's going to end up causing you, the PI, a huge amount of trouble when you are when you enter the study and someone says, so now we're going to assess this, and it says something different in different sections. This sounds trivial, but when you've amended a protocol, or when you start writing it today and then you take it, you finish it this week, you get all of our comments, you change it, and then you take it to your home institution and they change it, and then the IRB changes it. It's very easy to have sections missed, and so it's important to make sure that your primary objective, above all else, is consistent. It should be very clearly stated, and it should be based on a clear hypothesis, because this is going to be the basis for your statistical plan. And then the secondary endpoints, really do what you want, but just be prepared to collect all that data and to be careful about your data collection and make sure that you know exactly how you're going to store all of that data, because it's really compelling and exciting to think of all of the other data points you can get while you're doing this work, and in the end you end up with missed data that makes the whole endeavor useless. So the introduction section, a really good introduction section would be what you would submit to a peer-reviewed journal for a review article. And it's compelling enough that would be part of your grants application, that the people who are reading your protocol from the IRB, who are pediatric pulmonologists, are fascinated by whatever you're talking about for ALS or for Huntington's disease. It should be a really concise review that is pertinent to your topic and sets the stage for why this is a good idea. And you really want to be an educator in this place. And then you also want to be very clear in defining what is your intervention, what thing are you going to do, what are the known potential risks, what are the known potential benefits of this intervention, why is it justified that you're doing this? And all I can say is, sitting on these review committees, it's really nice if you don't make people work to try to understand why this is a worthwhile endeavor, if you can explain to them why it's a worthwhile endeavor. The study design is really your treatment plan. So if you could go back to your primary aim and then your secondary aims, and for each one of those say, to answer the question of does this impact overall survival, I am going to give aspirin at 1 week intervals, and I'm going to monitor that for 12 months. That's your study design. And then when you get into the other portions of the protocol, then you're going to say the aspirin has to be given in this form on this day, and I'm going to keep a drug log and all of those things. But this is really what you're saying. In order to address your primary objective, what are the things you're going to do? And you need that for each one of your objectives. So if you write 15 secondary objectives, then you have to have a study design for each of those secondary objectives. This is also where you'll have all the details of your dosing, your schedule, your route, your plan for changing dosing, your duration of intervention, duration of follow-up. So when you're in your groups thinking about these things, this is where this is going to go in the study design. Selection and enrollment of subjects is a critical point of failure for clinical trials, and so it's a really important time to think carefully about who you want to include. And broadly, there will be a lot of talk about this later in the course, but you want to define your target population by the idea is you want as homogeneous as a group as possible while making it feasible to actually enroll a patient. And then you want to be very specific. So too often people do the copy forward. They've copied from another protocol, and so they'll say patients have to have normal liver function tests. So then stop and ask yourself, is that really true? Do they have to have normal liver function test? Because what's going to happen for the random patient who gets an AST that's 800? Is that patient now excluded? And as the clinician, you'd sit there and you say, oh my God, that's ridiculous. This drug has nothing to do with the liver. But you put that in your protocol. So go carefully through each of your exclusion criteria and inclusion criteria, make sure that they're needed. So this is this is a phase 2 study I'm running now. This is our entirety of our eligibility criteria. It's 9 things, and it basically defines a newly diagnosed glioblastoma population. And all the things I've put in here to say this is who you have to be. This patient, you have to be newly diagnosed. You have to have had these 3 therapies. That's it. And then the exclusion criteria is that they can't be likely to have more risk than the average patient to be on this drug. So they can be relatively brief and specific. So then the next part of this, after you do your inclusion-exclusion, is to talk about what is your plan for patients? How many do you need? How many do you plan to enroll per month? If you're going to do a multi-center study, how many do you plan at each site over time? How are you going to monitor that? How are you going to get the feedback from the sites about how enrollment's going? How are you going to modify things when that enrollment's not going well? How are you going to identify patients? How will they be screened? Who's going to record that and where? And what will the consent process look like? All of that is under the enrollment of patients. And then the study interventions goes back to what we talked about before. The study design was what we planned to do. Now we're going to be very specific about our study interventions. On this day, plus or minus 3 days, at this place, infusion clinic, this person is going to get this drug. And so whatever it is, whether you say on this day someone's going to do half an hour of yoga, is it going to be Ashtanga yoga, or what is it going to be? You have to be very specific here. Does it have to be by a specific instructor, or can any certified instructor? And then any foreseeable complication, if you know a drug is likely to cause nausea, that's just what it does, then you have to talk about that and what you would propose as an intervention. So here's another place I really like schemas. I think this speaks to some of my learning disabilities, but I like to look at it. This, again, is one whole protocol on one page. This is what's going to happen to every patient who enrolls on this study. They're going to get this drug. They're going to get 4 weeks of rest. They're going to go to initiation cycle. Then they're going to go through this protocol. And that's it for every patient. The study interventions, you have to say what's a required intervention. So in order to remain on this protocol, you have to be doing these things. If you don't do these things for more than 4 weeks, you are no longer on treatment, for example. But you have to define that. And then what's prohibited? If you're getting a certain drug, you can't have surgery. If you're doing an exercise regimen, you can't enroll in a gym at the same time. Whatever it is that you need to prohibit to try to limit your confounding variables. The next section will be your clinical and laboratory evaluations, and there's a lot of language in here, a lot of verbiage, but this is the important table that the verbiage will then explain. And these are your table of required observations. So essentially, this is what you're saying. These are all of the data points I'm going to collect and when I'm going to collect them. And then you want to give yourself some time windows on this. So within 7 days of last dose, you have to get the off-treatment evaluation. And all of these numbers in here have some caveat listed below. So they'll say, have to be done on the MRI scanner that's a 3 Tesla, or can be done within 5 days, or whatever it will say. For each one of these things, you should be able to even make a case report form. So these are your data points that you are collecting. This is the data that you will be managing. So to think about that when you're making your table of required observations, and then think about what you're going to do with the data that you collect. You literally have to detail every test and measure, particularly, maybe not when you're doing an early exploratory study, but if you say it's very important that I'm going to assess liver function tests. How? By what assay? Which laboratory norm are you going to use? And you have to specify that, and then in your protocol you have to say that. If you're going to monitor peripheral blood monocytes after they've been exposed to some lipid-lowering agent, you have to specify what that protocol needs to be. Now, there's some room for variability here, because if you have a lot of very specialized techniques, in order to avoid a lot of amendments, sometimes it's best to have an operations manual in addition to your protocol. So you may say, we're going to do this fancy peripheral blood monocyte assay, see operations manual. So as the technique is adjusted, the assay is changed, you're not making amendments over and over. And then you want to also specify, a lot of studies will require a screening a set of laboratory to even find out if they're eligible, versus the on-study evaluations. And it's really nice to specify that in your protocol up front, so that you know that these ones are screening eligibility and these are actual study measures. So adverse events. It depends on who you're writing the protocol for. There is a lot of highly developed language about adverse event reporting, and oftentimes it's best to work with whoever your sponsor is, or whatever agency you're likely to be communicating with. For example, the NCI has a very specified adverse event reporting format. But essentially, this is any unfavorable, that's the definition there, I won't read it, but anything that happens that you don't want to happen, even if it's not related to your drug. If a patient is on your study, if they've signed consent and they crash their car and get a laceration on the head, you have to report that they crashed their car and got a laceration on the head, and that it is unlikely related to your drug. Now, you can write your protocol to say, I'm not going to include those things, but then you have to be prepared to justify that. And then you have to list all of the expected and anticipated adverse events, particularly with repurposed drugs. We know a lot about these drugs. We know exactly how often people develop diarrhea. So we can say 30% of the time patients are going to develop diarrhea, and this is what I would propose to intervene with that. And if they develop grade 3 or grade 4 diarrhea, we can talk about grading later, then they have to stop drug for this long. So you give all the contingencies here. This is why I'm always looking at my protocol. What do I have to do if they have grade 3? For how long? When do I stop that? And it says what to do. Again, you define your grading criteria, and it may be industry, it might be division. And then you have to say who's going to hear about this AE. So for serious AEs, your IRB for sure has to hear about it. Often your sponsor has to hear about it. Sometimes the FDA has to hear about it. And so knowing those up front and having your team know those up front. So the minute the patient calls in and says, I was in the ER yesterday, you know what pathway to activate in terms of reporting. That's in your protocol up front. So again, we have to list the required versus suggested responses to adverse events. Some of these are very highly regulated for you already, and you borrow the language. But here's an example. A grade 3 hemorrhage, the protocol will say no more drug. Versus grade 2 nausea, you might have some suggestions about what antiemetic drugs you would use. What are your criteria for stopping treatment? And then what's your time period for following patients once they've had an adverse event? How long are you going to follow them until that's resolved? An important distinction, and I won't read all of this, but this is also in your slides, is to make the distinction between off-treatment and off-study. And so when you're writing this part of your protocol, to think about that. Off-study means you are no longer collecting any information about this patient. That in order to get new information about them, you would have to get a new consent form, you'd need a new HIPAA waiver. Off-treatment means you are no longer doing the intervention, but you're still following that patient for data, contacting the patient, collecting their medical records. And specifying the criteria for each of those is important. Statistical design is one of the really beautiful things about this whole course is how deep your knowledge of statistical design will be when you're done. But essentially what this portion will break down to is the statistical design is a summary of the statistical plan that should be running through your whole protocol. This, a beautiful protocol is built with the statistician from the start, your partners in designing that protocol. And so that will show up in your introduction, it will show up in your objectives. But again, it's really critical that your objectives are reflected here exactly how you reflected them at the start of your protocol. And then there's additional language here about the statistical rationale. So why did you use an adaptive design? And what were the assumptions made to allow that? And what were the assumptions for the sample size calculation? And all of that stated very explicitly. Based on a history of a 10% known bleed risk after aspirin, we assumed that we could use a sample size of 70 patients to see a 5% effect size. And then you'll give the reference for where you made those assumptions. And then a separate section will be on your data analysis plan. And some of that is your predicted data analysis plan, and then for all the secondary objectives in general, you have a more broad leeway on what that analysis plan will look like. Just as important is your plan for data monitoring and reporting. That started back with our table of observations where we said these are all the data points we're going to collect. So as soon as you're collecting something, you need a place to put it. And so now you're saying, what's my database going to look like? Who's responsible for that database? Who has access to that database? How are patients coded? What's the data management plan? Can I do that all myself? Do I need institutional oversight? Do I need a data safety monitoring board? And that will all come about depending on the risk and complexity of your study. And then what's your plan for data sharing? Who's going to get the data? Are you holding the database? Is your sponsor holding the database? When is it locked? Who has access to it? When is it unblinded? And then all of your quality assurance, so all of my alphabet soup down there, is that the quality assurance can be done just by the PI. Of course, the PI is highly biased, so that might not be ideal. It can be done by a clinical research office, either a hired clinical research office or an institutional clinical research office. It can be done by a data safety monitoring board, but what level of quality assurance are you requiring for your study? And then what's your schedule for audits? And it's ideal if you set that up front, that we're going to do an audit after the third patient is enrolled and then after every sixth patient after that. Or we're going to do an audit at once 50% of the patients have reached the primary endpoint. Protection of human subjects, it's interesting that this comes at the end of protocols to me because it's kind of where people peter out in their energy level, and it perhaps should be the first thing we think about. This is really where you're going to say, I recognize I'm putting patients at risk. I'm going to do the following measures to mitigate that risk. I'm going to share my plan as transparently as possible with the following organizations, and I'm going to tell patients about this risk in this way. This is how I'm going to explain it to them. So then you're going to say how often you're going to check back in about changing that risk, because let's say you're using a drug that was repurposed from some kind of oncologic indication, and now the oncologic field has figured out there's a new risk. How are you going to modify how you report that risk to all of the stakeholders? And then what's your process for consent? How are you going to guarantee that patients really went through informed consent? And it's a hard work to just get your protocol done, but ideally you'd be writing a consent form at the same time. That would be your model consent form. And that is the abrupt end. So that's basically the key portions of the protocol that when you open up the prototype that's on your disk, that's what you're going to see with some suggested language to put in there. If there are things in there that you don't understand, if you say, what's a da-da-da-da, you probably don't need it. If there are things in there, if you come to the end of writing your protocol and you say, I have no idea how I'm going to handle X, then you should probably invent a subsection that's about X, because every protocol is not the same. Things will have different needs, but everything you're going to do in your study has to be documented in your protocol, so that if you quit tomorrow and you're now, a character at Disney World instead of doing what you do now, someone else can carry on that protocol without any pause. And the reason for that, just to tell you the rationale of that, once you enroll one patient on a protocol, if you stop that protocol early, then everything that patient did on that protocol, all the risks they took, all the time they gave you, was for nothing. So you want the protocol to proceed as clearly and as seamlessly as possible so that every patient's contribution is counted. And that's what I got. Great.
Well, that was great. A whirlwind tour. One thing that's often easy for us to overlook, especially in a context like this, is the most important thing that she said is that really you have to go out of your way to protect human subjects. I mean, it really is a tremendous privilege to have these patients trust you that you're going to enroll them in a study and you're not going to hurt them. And that's exactly the same as if you're going to get them through a course of clinical treatment or a tough illness or a major surgical procedure. We tend to take it for granted in this, but you have to keep that in mind. What she basically talked about is the core of clinical research. It's how you design your study. Everything was right there in less than 30 slides. And all the rest of this is all the other ancillaries that go with that. Now, I realize that at your stage, you're all trying to figure out how to do this. There's this tendency to focus down on the details. You know, you're all very smart, you're all very hardworking, and I'm sure you memorize your biochemistry books and your anatomy books and your physiology books, and you'll memorize this too. But you can't lose sight of the fact that the most important thing is get a good hypothesis, get good specific aims, because if you don't have that, the rest of it doesn't matter. And it's really kind of a shame sometimes. You see someone go through a great statistical analysis plan, data sharing plan, data collection plan, and yet the whole thing is flawed at the foundation. It doesn't have a chance to fly. You know, many of us in this room review grants for study section and for various foundations. And it's kind of interesting, at least the way I do this, you start glancing over these things to get the lay of the land. And oftentimes, specific aims are bad, you know, forget it. But if they're good, you say, okay, this is really good. You know, this could be an interesting study. And you start thinking, how would I do this? What's the best way to design this? But then it falls apart because you haven't paid attention to the protocol, which is basically your research plan. And you start realizing that even though they're asking good questions, the way they're doing it is wrong. The way they're collecting patients, the way they're randomizing, their inclusion or exclusion, the way they're going to analyze the data. So you have to strike this balance between big picture and details. A couple of other key points that she mentioned also is that, you know, just imagine if you're doing a small trial at your own place, pick an intervention, you know, say stroke or multiple sclerosis or MS or myasthenia, whatever. And what if you're out of town? Well, you'd ideally like that to continue, right? So maybe your partner or someone else can enroll patients in your study, and if you have a well-specified protocol, then that's great. You continue to enroll patients while you're traveling. Well, what if they start doing things differently than you do? You get two docs together, they're not going to do things the same way, right? Well, then what if suddenly you get 2 centers or 4 centers or some of these big 50-center phase 3 trials? And as Jaishri said, it's just amazing how much variability there can be. And if you're a PI on something like that, it's, you know, you're working 68 hours a week, dealing with enrollment, dealing with a lot of dispute resolution and things like that. So you can't overestimate the importance of dealing with this up front. And finally, she made a great point a couple of times about schemas and graphs. Again, it's partly to help you conceptualize it, but keep in mind, you're going to try to get this funded by somebody. And as someone who reviews these for funding agencies, it's a lot easier, a lot cleaner to have a visual than try to wade through paragraphs of black ink on white paper. Well, I think that's all I have for now. We already have a question.
No